Hello everyone, welcome back to my channel all about life sciences. In today's session, I'll be teaching one of the most important staining techniques of microbiology called as gram staining. But before that, please like, share and subscribe to my channel and also hit the bell icon for further updates. Are you guys ready? So let's begin. What is gram staining? Gram staining was discovered by a Danish physician, Dr. Hans Christian Gram, in 1884. It is a type of differential staining in which we differentiate two major groups of bacteria, namely gram-positive and gram-negative bacteria. What is the principle of gram staining? Gram staining is based on the principle of cell wall components. For example, in gram-positive bacteria, the cell wall is thick and composed of about 70% of peptidoglycan. Peptidoglycan is a polymer of sugar and amino acids that form a cross-link-like structure. This cross-linking helps the stain to remain entrapped in the cell wall and not get affected by the action of ethyl alcohol. Whereas in gram-negative bacteria, the cell wall contains less amount of peptidoglycan and high amount of lipids. The presence of lipids will get dissolved by the action of ethyl alcohol. As a result, large pores will develop in the cell wall and helps in the leaking of the main stain, due to which these bacteria acquire the counter stain and appear pink or red in color. As you can see in this diagram, the gram-positive bacteria contain large amount of peptidoglycan layer, whereas in gram-negative bacteria, the peptidoglycan is thin and the layer of lipid is thick. Now, what are the reagents required for performing gram staining? First, crystal violet. Crystal violet act as a main stain. Second, grams iodine, which act as a mordant to fix the main stain. Next is ethanol or alcohol, which act as a decolorizing agent. And last is the safranin, which act as a counter stain. Now let's take a look on the procedure of gram staining. First, prepare the bacterial smear on a clean glass slide and heat fix the slide. Then place few drops of crystal violet or the main stain and wait for about 20 seconds. Remove the excess stain with flowing water. The bacterial cells are now stained. Add the grams iodine and wait for a minute. Then pour off the excess iodine and place the slide in a beaker containing alcohol or ethyl alcohol. Now gently rinse the slide with water and cover the slide with the safranin for 20 seconds. After that, remove the stain with water and blot dry the slide and observe under microscope. Observation. Some of the bacterial cells acquire the main stain and appear blue colored, whereas other bacterial cells take the counter stain and appears pink. Now, let's take a look on the result of gram staining. Purple or blue colored cells are gram-positive bacteria, whereas pink or red colored cells are gram-negative bacteria. Summarization of gram staining. In this figure, you can see the reagents, time required for reagent action, and the color of gram-positive and gram-negative bacteria. First is smear, which is initially colorless in both gram-positive and gram-negative bacteria. Then we add crystal violet for 20 seconds. The gram-positive bacteria appear purple. Similarly, gram-negative bacteria also appear purple in color. On adding grams iodine for 1 minute, gram-positive bacteria appear purple. In the same way, gram-negative bacteria also appear purple in color. By adding ethanol for 20 seconds, the gram-positive bacteria shows purple in color, whereas the gram-negative bacteria appear colorless. And on adding the counter stain or safranin for 20 seconds, the gram-positive bacteria appear purple and gram-negative bacteria appear red in color. Therefore, the gram-positive bacteria appear purple in color, whereas the gram-negative bacteria, which takes up the counter stain, appears red in color. So students, this is all about gram staining, and about gram-positive and gram-negative bacteria we will discuss in the next video in detail. Till then, if you have any doubt regarding the topic, do comment below and I'll try to solve it out. And don't forget to like, share and subscribe to my channel.